My name is Stephen Kohler, and today I'll be talking about the seasonal dynamics of mercury in the northern Barents Sea. I just received my PhD 1 month ago, so the doctor title feels a little new. Anyway, um, let's jump into it. So, for my work, I focused on the seasonal cycle of mercury in the pelagic water column, and here I've constructed a simplified seasonal cycle from what we know from literature so far. With atmospheric mercury depletion events occurring in the early spring, we have river and erosion inputs shortly after. We can also have additional mercury inputs from sea ice melt, as well as exchange with the atmosphere throughout the summer months. And also within the water column, we can have mercury scavenged with the sinking biological particles and in situ production of methylated mercury forms. But if we categorize all the Arctic mercury water column observations that we have to date, to the best of my knowledge, only 97% of them occur only within these 4 months of the year between July and October. So the main focus of my work and the big overarching question was, is there a seasonal cycle of mercury in the Arctic water column? As we know that mercury in the atmosphere and the riverine inputs, etc., follow a seasonal pattern. And this is in addition to the seasonal light regime and biological activity. So this work was done in the northern Barents Sea on board Crown Prince Haakon during 4 seasonal cruises in August, December, March, and May, which you'll see later represented as summer, winter, late winter, and spring. We collected seawater for total and methylated mercury. We collected some cryosphere samples when possible, and surface sediment as well for total mercury analysis. Our station sampling map is here in the northern Barents Sea. This consisted of 7 stations from P1 to P7. And here's also a simplified oceanographic scheme of AW or Atlantic water, as well as PW polar water. And the mixing between these 2 water masses can also give rise to modified Atlantic water, as well as warm polar water. For our field sampling, we used GoFlow bottles. We aimed for 8 depths per station. We manually deployed them attached to a Kevlar line, as you see here in the video. This is from May of 2021 cruise. We sent them in closed to try and avoid contamination from both the surface ship as well as the atmosphere. When we brought them on board, we transported them to our homemade onboard custom clean lab, where we sampled unfiltered seawater for total unmethylated mercury. We measured our total mercury by CVAFS and our preserved methylated mercury via GC ICP-MS. And just note that the methyl mercury concentrations you'll see presented are the sum of both dimethyl and monomethyl mercury. So 2 results have already been published from this work, which you may or may not have read. The wintertime mercury concentrations on the shelf, as well as the distribution pattern of mercury in the sediment. But the 3rd manuscript, which is still in review and revision, which I would like to talk about today, is the last one, which is biotic transformation of methyl mercury at the onset of the Arctic spring bloom. So to put it into focus, we have our seasonal cycle. Our first 2 cruises, which were already published, were from August and December. But the rest and the majority of today's talk will focus on these last 2 cruises in March and May of 2021. So our results for our transect, we have our southernmost station P1 in the left of our ODV plot and P7 on the right. The top 2 plots show total mercury with late winter on the left and spring on the right. In late winter, our total mercury concentrations for the entire transect were 0.56 picomole per liter, while in spring, our average total mercury is 0.69 picomole per liter, with the ranges between 0.4 to 1.6 in late winter and 0.46 to 2.37 in spring. If we just look at the plots, we can see that there appears to be some elevated surface total mercury during the springtime, as well as in deeper waters in our basin stations, P6 and P7. In methylmercury, which are the two middle plots on the left and the right, in late winter, our average in femtomolar is 112 plus or minus 76. Whereas in spring, we have a decrease to 69 plus or minus 75. We can see a decline of our methylated mercury concentrations on our shelf stations, which is P1 to P5. These are the shallow stations. The bathymetry is in black, in case anyone was wondering or if I missed that. And our bottom plot shows the percent methylmercury as a function of the total, where we can also see this decline in percent methylated mercury with approximately 20% of THG in late winter and only about 10% of total mercury in spring. So first, we want to look at the water masses for our two cruises to see if any changes observed in our mercury concentrations are just due to a change in the water mass, where we again have P1 on the left and P7 on the right for each plot, and then March is shown on the left graph and May on the right. For most of our shelf stations, polar water in the light blue represents most of our depths, including the upper water column. P1 has that influence of Atlantic water and modified Atlantic water. And the West Spitsbergen current, which crosses perpendicular to our transect, appears here at P6 and P7 for both cruises. We also have some pockets of warm polar water at our shelf stations, P3 and P4. And there's another paper, Van Angelen L 2023, which shows that there can be an incursion of a bit of the West Spitsbergen current into this area. So if we divide or categorize our total methylated mercury by water mass, here we have total mercury on our left with the late winter cruise, LW, and SP, spring cruise. We can see some differences between the average concentration, and the same for our methylated mercury species with late winter and spring, and we can see this decline. When we perform statistics on this analysis using log transformation, there is a significant difference in the total mercury concentration in both modified Atlantic water as well as polar water. And for methylated mercury, we do see a significant difference between our polar water methylated mercury concentration. And as polar water makes up the majority of our stations, this could suggest that there's a different processes at play to explain our decline in our methylated mercury concentrations. So the environmental conditions, this is just one station, but here P3, this is the fraction of sea ice coverage that we observed during our cruises. Our late winter cruise is here in blue, and our spring cruise is in yellow-orange. And we can see a large change in the fraction of sea ice coverage in between our cruises. And as we saw, our total mercury concentration is a bit higher in polar water. So this could suggest some input from AMDEs or ice melt during this time frame. Our under ice water samples collected during both cruises also help reflect this hypothesis. where our late winter total mercury is 0.58 plus or minus 0.02 picomole per liter, and then spring it's up to 1 picomolar. We also saw the beginnings of the spring bloom along our cruise transect, represented by increasing standing stocks of most of our various types of phytoplankton at P1 up to P6. So regression analysis in the upper 500 meters of our water column with 95% confidence interval. We have our late winter data points in blue and our spring data points in yellow. We do see significant relationships with both total and methylated mercury with particulate organic carbon. We see a positive relationship with total mercury and a negative relationship with methylated mercury. So this suggests that THG is associated with the particulate phase. So even though we have an increase in total mercury concentration, we could also have an increase in the percent of particulate mercury. Chlorophyll A shows a similar pattern for total and methylated mercury, where we more or less have significant relationships of total and methylated mercury in the springtime in the yellow plots. We do have one in late winter, but as chlorophyll A is virtually non-existent, I'm not so confident in this relationship. So this again suggests that the total mercury is associated with phytoplankton, as expected from POC. So lastly, we have the Hg-POC ratio against chlorophyll A, and we see significant negative relationships. So we see a declining ratio as our chlorophyll A increases. So because both THG and methylmercury have a lower ratio with increasing chlorophyll A, this could suggest that mercury is being taken up during this time period. But if methylmercury behaves like total mercury with regards to its chemical speciation or association to the particulate phase, could there be biotic demethylation occurring during this uptake? But first, we have to rule out some of the other changes in our methylated mercury concentration. So we integrated the water column between late winter and summer and calculated delta methylmercury, or net change between our two cruises. And we tried to quantify photodemethylation, potential scavenging, evasion, and we attributed the rest as dark demethylation. So we used satellite PAR, percent over water, vertical attenuation, and literature photodemethylation rate constant. For photodemethylation, scavenging, there are POC fluxes available from these cruises, and we did estimate particulate monomethylmercury from KD and POC, as well as estimated TPM. And lastly, we also estimated potential dimethylmercury evasion from open water, wind speed, and an estimated DMHG concentration in the primary mixed layer. We saw that the water masses don't change too much between the two cruises, so we've ignored advection for this percent delta MEHG, and we attributed the rest as dark demethylation or biological demethylation by both phytoplankton and bacteria for these two cruises. So we could have both uptake and demethylation by phytoplankton and or bacteria during our transition to our spring cruise. To put it all together, we have now our 4 cruises. These are total mercury from late winter, spring, summer, and August. It's in the same transect each time. There is 1 year in between because of COVID. But we can see some patterns emerging where we have our lower concentrations in our wintertime periods, as well as our increase in surface waters during spring and summer. We have potential mercury inputs during the spring, and we have some more stratification in the summer, as well as continuous exchange with the atmosphere. So we integrated both total methylated mercury in the upper 100 meters for each station, and we begin to see this seasonal pattern start to emerge. We see a rise in total mercury during the summer months, as well as a decline during winter. And we see this decline to our spring cruise and an increase towards the late summer and going into the polar night. So to try and evaluate the drivers between all four cruises, we performed PSA analysis with other potentially relevant environmental parameters. And for our spring and summer cruises, we can see total mercury concentrations more or less clustering with biological parameters such as PUC and chlorophyll A. And this is the same for the summer cruise, where we have THG with PUC, bacteria, and chlorophyll A. And this bacteria, as we have a shift to a more oligotrophic community or heterotrophic community during summer, this may help explain this clustering over here. And they are opposite to a lot of the depth-related parameters like nutrients, salinity, etc. So when we go back to our seasonal cycle for this work, we plotted the general trend of total mercury as a function of the mercury seasonal cycle. And we can see that our total mercury concentration is quite representative of the mercury inputs during the early spring, the summer, and also in our previous paper, we highlighted that scavenging may play a potentially important role. Same for methylated mercury. We plotted the general trend of the seasonal cycle, where we see this increase throughout the summer months, most likely driven by this in situ methylated mercury production, slight demethylation during the polar night, and as we suggested in this third and final paper, potential methylmercury uptake and subsequent demethylation in both surface and deeper waters as well. Yes. But in general, the project, we contributed more data for the four seasonal time periods, as well as data in surface sediment. There is more data coming, mercury data from this project, the Nansen Legacy, in zooplankton and in fish from also these same four cruises, as well as the environmental parameters from the cruises, which are now being published in a new special issue in Progress in Oceanography. But I suggest that for our future work, we should measure additional mercury species like the gaseous forms and the particulate forms to help us better quantify some of these processes. And now that many of the environmental parameters are available, I suggest a model to try and model mercury species from a seasonal perspective with all these environmental drivers, and potentially to incorporate more seasonal studies to evaluate the Barents Sea and other Arctic shelves if they behave in a similar manner. So thank you for your attention. This is a nice figure that one of our artists from the project has created, which we're still tweaking, I would say, but it's what we propose as a seasonal mercury cycle in the Arctic marginal ice zone. So that's all I have for today.